Good morning to all of you. I am Dr. Nikunj Patel from Department of Biochemistry, Smimir Medical College, Surat. In today's lecture, I will discuss about the general reaction of amino acid, that including transamination, deamination, transmethylation, and transsulfurization. In this lecture, we mainly focus on the transamination and deamination. All of you are very well known about the basic structure of the amino acid. Every amino acid contains the alpha carbon. Along with that, for group 1 it is a carboxyl group, one it is amino group, third side it is occupied by the hydrogen, and fourth it is a side chain. In the different amino acid, different side chain are the present. Depend on that, there can be classified as a different group of amino acid. So amino acid have a that nitrogen. So major source of the nitrogen in our body, it is come from the amino acid, remember. Basic reaction related to amino acid metabolism, that includes reaction that is due to the carboxyl group. One of the reaction related to carboxyl group, it is a removal of carboxyl group. That reaction, it is known as a decarboxylation. So one of the example of the decarboxylation, it is conversion of the histidine to the histamine. In that case, what happened? That carboxyl group, it is removed in the form of the carbon dioxide, and itself it is converted into the respective amine, because that amino acid contain the two main groups. One it is a carboxyl group, one it is amino group. Carboxyl group, it is removed. Now only amino group are the present. That's why it is known as a amine. Okay, histamine. So by that, various amino acid, it is converted into the respective amine. That enzyme required for reaction, it is a decarboxylase. It is a pyridoxine dependent enzyme, means active form of the pyridoxine in the form of the pyridoxal phosphate. It is act as a cofactor for that enzyme. You have to remember that PLP, it is required for the decarboxylase. Whenever you learn the pyridoxine vitamin, the various reaction required for catalyzed by the pyridoxine, one of the reaction, it is a decarboxylase. So you can correlate with that, same like with the amino acid metabolism and vitamin metabolism. So in case of the vitamin, the pyridoxine vitamin, one of the metabolic reaction in the pyridoxine metabolism, it is decarboxylase. So in generalize, you have to remember that decarboxylase enzyme required for the decarboxylation, and that require the cofactor, it is pyridoxal phosphate, and it convert the amino acid into the respective amine. Like histidine, it is converted into the histamine. Tyrosine, it is converted into the tyramine. Tryptophan converted into the tryptamine. All are the amine. Okay. Lysine, it is converted into the cadaverine. It is also diamine. Glutamic acid, it is converted into the GABA. So remember that by the help of the decarboxylation, that amino acid, it is converted into the respective amine. Second major reaction related to the amino acid, it is transamination, means that reaction, it is related to the amino group. First reaction, it is related to the carboxyl group. Now that reaction, it is related to the amino group. Related to amino group, mainly two reaction are we have to study: transamination and deamination. Name itself suggests that trans means transfer. So in case of the transamination, amino group does not release in the free form, but it is transferred from the one group or one molecule to the another molecule. So in case of the transamination, what happened? That amino group from the one amino acid, it is transferred to the keto acid and produce the new amino acid. Okay, what happened in that case? Amino group from the one amino acid, it is transferred to the alpha keto acid and it produces the new amino acid. So amino acid that donate that amino group itself, it is converted into the keto acid, because amino group it is removed, so only carboxyl group are the present, so it is converted into the keto acid. And one molecule that receive the amino group, it have already keto group, it receive the amino groups, now it is converted into the amino acid. So it is a pair reaction, one amino acid and one keto acid converted into the one new amino acid and one respective keto acid. For that reaction, enzyme it is required that is known as a aminotransferase or transaminase. Purpose of the transamination. What is the basic purpose of the transamination? First, it is a synthesis. First major purpose of the transamination, it is a removal of the ammonia. Okay, means it is ultimately removed from the one amino acid and transferred to the another amino acid. So it is also used as a channel the ammonia to the glutamate. Overall, from the all the amino acid, that ammonia it is finally channeled to the glutamate by transamination. Second, synthesis of the non-essential amino acid like alanine from pyruvate, aspartate from oxaloacetic acid. They are the synthesized with the help of the transamination. So synthesis of the non-essential amino acid are the major things. Third, it is also required for the gluconeogenesis. Various amino acid are the glucogenic substance like alanine. Alanine it is converted into the pyruvate by transamination. Then pyruvate it is converted into the oxaloacetic acid by pyruvate carboxylate, another enzyme, and it is enter into the gluconeogenesis. Same like aspartate converted into the oxaloacetic acid by transamination and it is channeled into the gluconeogenesis. So basic mainly three purpose, that is a channel the ammonia group to the glutamate, second synthesis of the non-essential amino acid, and third it is a required for the gluconeogenesis. Means glucogenic amino acid, it is through the transamination it provides the substrate for the gluconeogenesis. Enzyme in the transamination are the known as a transaminase, or aminotransferase. Two major examples of the transaminase are the AST and ALT. Alanine transaminase and that is a aspartate transaminase. That is also known as a SGPT, serum glutamate pyruvate transaminase, serum glutamate pyruvate transaminase, or that is also known as a SGOT, serum glutamate oxaloacetate transaminase. Okay. Its basic reaction, it is catalyzed by the AST or also known as a SGOT. It is a reversible reaction. Remember, one keto acid means oxaloacetic acid, it is a keto acid, along with the one amino acid glutamate. With the help of the enzyme AST, it is converted into the one amino acid aspartate and one keto acid alpha ketoglutarate. Okay, so oxaloacetic acid converted into the aspartate, along with that the glutamate converted into the respective keto acid alpha ketoglutarate. It may be reversible. Alpha ketoglutarate along with the aspartate can convert into the oxaloacetic acid and the glutamate. So that oxaloacetic acid can use for the gluconeogenesis. So that is the reversible reaction. Depend on the substrate availability, enzyme can catalyze the reaction in that direction. Same like the alanine. Alanine amino acid, it is converted into the pyruvate, then it is used for the gluconeogenesis. Okay. Or pyruvate may produce the alanine, that is a non-essential amino acid. So the reaction catalyze in the either direction, depend on the availability of the substrate. So always remember the one amino acid, that is a amino acid, one keto acid, that is alpha ketoglutarate. It receive the amino group from the alanine and convert into the amino acid, and alanine donate it amino group, so it is converted into the keto acid. So in majority of the all the amino acid catabolism, that amino group from the amino acid, it is a finally channel into the glutamate, because in that case majority of the amino acid catabolism, that one pair it is always act by the glutamate alpha ketoglutarate. So glutamate it is receive the amino group from the amino acid and it is in the form of the alpha ketoglutarate. Alpha ketoglutarate along with the one amino acid that channel into the glutamate and the respective keto acid. Also remember that both enzymes are the occur in the liver mainly. The AST are the mainly present in the cytoplasm as well as mitochondria in the cell. That can be present in case of the ALT, that is only present in the cytoplasm. ALT and AST. So ALT only in the cytoplasm, that is cytoplasm plus mitochondria. That is mainly present in the liver. In the some amount it may be present in the kidney and other organ, that is also present in the liver, okay, placenta, bone and other tissue. It is mainly increased in the liver damage. That is also increase in the liver damage, pregnancy, bony injury. AST it is a specific for the alcoholic liver damage, remember. And ALT it is specific for the viral hepatitis. Both are the non-functional enzyme in the blood, means whenever they are the mainly intracellular enzyme. So their function are the mainly in the cell, in the cytoplasm as well as in the mitochondria, depend on the enzyme. When cell are the damage from cytoplasm and mitochondria, it is released into the blood circulation, and in the blood the level are the increase, that can be suggestive of the that particular tissue are the damage. But in the blood they are the non-functional enzyme. So it is only used for the diagnostic purpose in case of the blood measurement. Various amino acids that not undergo the transamination include proline, hydroxyproline, lysine and threonine. That 4 amino acids are the mainly not undergo the transamination. They are the metabolized in the different way. Means that amino group are the metabolized in the different way. Otherwise, all major amino acid are the first channel that amino group into the glutamate. Then glutamate undergo the oxidative deamination and release that ammonia. Second major reaction related to the amino group, it is a oxidative deamination, means deamination. It may be oxidative or non-oxidative. Oxidative deamination means along with the deamination, one reductive reaction are the occur. In case of the non-oxidative deamination, the non-reductive reaction are not occur along with that. Oxidative deamination, it either may use the NAD, FAD or FMN, depend on the different enzyme. So major oxidative deamination are catalyzed by the enzyme that is known as the glutamate dehydrogenase. That enzyme either use the NAD or NADP. Remember, that is a peculiarity of that enzyme. That enzyme are the only present in the liver, in the mitochondria. That's why oxidative deamination only occur in the liver. That's why urea cycle are the occur in the liver, because that is the only reaction by which ammonia, free ammonia are the released from the glutamate, and that ammonia are the further enter into the urea cycle for the conversion of the ammonia into the urea. And enzyme are the occur in the liver, mitochondria. That enzyme has a second peculiarity is same enzyme either utilize the NAD or NADP variant. Reaction at the bidirection depend on the substrate availability. Glutamate convert into the alpha ketoglutarate and release the ammonia, or alpha ketoglutarate that accept the ammonia and convert into the glutamate. That enzyme are the inhibited by high level of the NADH, NADPH or ATP and activated by reverse thing ADP. It is activated by ADP. So remember that glutamate dehydrogenase, it is a major enzyme or most important enzyme for the oxidative deamination that present in the liver mitochondria that utilize the NAD and NADP both reaction. It is a bidirection depend on the substrate availability, inhibited by the NADH, NADPH and ATP mainly and activated by the ADP. Second oxidative deamination that utilize the FMN is L-amino acid oxidase enzyme that convert the remove the amino group from the L-amino acid. Third, it is a D-amino acid oxidase, same like the L-amino acid oxidase, D-amino acid plus FAD, it is converted into the respective keto acid plus ammonia are the remove and FAD converted into the FADH2. So all three are the oxidative deamination, because along with that one reductive reaction are the occur. Second group of reaction, it is non-oxidative deamination, means without undergo the oxidation of the amino acid. Various amino acid are the undergo the non-oxidative deamination like sulfur containing amino acid, mainly cysteine and homocysteine undergo the oxidative deamination. OH group containing amino acid, serine and the threonine and histidine. The 5 are the major amino acid that undergo the non-oxidative deamination. How you remember? Okay, OH group containing amino acid, sulfur group containing amino acid and histidine. So mainly two things you have to remember. Amino acid are not undergo the transamination like lysine, threonine, proline, hydroxyproline, and amino acid are undergo the non-oxidative deamination like cysteine, OH group containing amino acid and histidine. Second important reaction related to amino acid metabolism, it is transmethylation. Transmethylation means transfer of the methyl group to synthesize some important product. Major donor of the methyl group in the transmethylation, it is a SAM, S-adenosyl methionine. That intermediate are formed in the methionine metabolism, that is a SAM molecule. Okay, it is synthesized in the methionine metabolism. Methionine plus ATP in the presence of the adenosyl transferase enzyme, the adenosyl group from the ATP, it is a transfer over the methionine and produce the S-adenosyl methionine. Now that group, it is a transfer that methyl group, okay, methyl group to the various acceptor molecule and produce the methylation. So that is a main reaction. As adenosyl methionine as a donor of the methyl group, one it is a acceptor of the methyl group molecule. When react, that produce the methylated product and that itself as adenosyl methionine, it is converted into the as adenosyl homocysteine molecule. So that is a transmethylation reaction. Methionine, a donor of the methyl group to the acceptor of the molecule. So various molecule are the undergo the methylation reaction are non-epinephrine that produce the epinephrine. 1-amino acetate produce the creatine. Ethanolamine by trimethylation, it produce the choline. N-acetyl serotonin that produce the melatonin, it is a neurotransmitter. Carnosine produce the by methylation anserine, it is present in the muscle tissue. That is amino acid are the present in the one type of the amino acid intermediate metabolite are the present in the muscle tissue. So remember the various reactions that undergo the transmethylation. So creatine by the transmethylation, choline by the transmethylation, melatonin by transmethylation, anserine by transmethylation, and epinephrine by transmethylation from non-epinephrine are the example of the transmethylation reaction. Transsulfurization, same like the methylation, transfer of the sulfur group from one molecule to another molecule. It is ultimately not a directly transfer of the sulfur group, it is a series of the reaction by which methionine, it is a sulfur containing amino acid, converted into the cysteine. Okay. So in the series of the reaction, what happened that methionine contained the sulfur, it first converted into the S-adenosyl methionine, then in the series of the reaction it produced the S-adenosyl homocysteine, then homocysteine along with the serine, it produced the cystathionine, it is an intermediate of reaction that require the enzyme, it is known as a cystathioninase synthase. Cystathionine synthase. And most important things, it is a PLP, it is a cofactor for that enzyme. In the second step of reaction, cystathionine hydrolase, okay, or cystathionine nase enzyme catalyzes cystathionine into the homoserine and L-cysteine. So now sulfur, it is entered into the cysteine group. So cysteine, it is a synthesized from the methionine, that's why it is a non-essential amino acid. And what happened in that case, sulfur present in the methionine, it is transferred to the cysteine. That's why it is known as a transsulfurization reaction. So it is a series of reaction by which cysteine are the synthesized. So why cysteine it is non-essential? Because it is synthesized from the methionine. And whenever the defect occur in the enzyme of the transsulfurization, that produce the homocysteinuria and cystathionine, because when defect occur in that enzyme that ultimately increase the level of the cystathionine or homocysteine, both are the increase and ultimately excreted in the urine and gives the homocysteinuria or cystathionine. Sometimes that cofactor pyridoxine deficiency may affect the enzyme activity, and that also may lead to the increase the homocysteine level in the blood and urine. Now, the source of the ammonia. We have already discussed about the source of the ammonia, that transamination followed by the oxidative deamination, it is one of the major source of the ammonia. Okay, like aspartate first donate that ammonia group to the alpha ketoglutarate and produce the glutamate. Then glutamate undergo the oxidative deamination by the dehydrogenase enzyme and produce the ammonia. Direct oxidative deamination means like L-amino acid and D-amino acid are undergo the direct oxidative deamination. Non-oxidative deamination like serine, threonine, cysteine, histidine are the undergo the non-oxidative deamination, and various non-amino acid source like purine, pyrimidine, various amine like putrescine, cadverine, biological amine are when catabolized it produce the non-amino acid. Urea itself in the intestine by urease enzyme it produces the ammonia. So they are the various source of the ammonia in our body. Why we learn about the ammonia? Because basic normal level of the ammonia, it is in the blood around less than 50 micromole per liter. Even it is increased up to the 100 micromole per liter, that might be leads to the unconsciousness. And when it is increased up to the 200 level, it may lead to the convulsion and coma. So it is, remember, it is very toxic to our neurons. Various form of the ammonia, that's why whenever the ammonia it is released from the amino acid, it is immediately captured by another amino acid or another molecule, and free ammonia level might be in less than 50 micromole per liter. So glutamine that captured the ammonia in the form of the glutamate plus ammonia. Glutamate, it is formed by the alpha ketoglutarate plus ammonia, and certain amount of the ammonia can transfer as in the form of the free. In the cell as well as in the blood, that ammonia are the equilibrium with the ammonium ion. Remember, around 99% in the ammonium ion and 1% as a free ammonia. So they are the various transport form of the ammonia, is a free form, glutamate form, glutamine form, Other form are the asparagine, means aspartate plus ammonia, alanine are the mainly from the muscle tissue. Amino acid in the muscle, okay, transfer the amino group from the muscle to the liver for the urea synthesis are the mainly in the alanine form. Amino group from the brain tissue mainly transfer in the form of the glutamine. Major fat of the ammonia, it is a urea in human. Certain animals can directly excrete the ammonia, like mainly aquatic animals, and certain can convert the urea into the uric acid, but in the human mainly it is excreted as a urea. Ammonia are the mainly excreted as a urea, and that occur only conversion of the ammonia into the urea are the mainly occur in the liver. So urea cycle only occur in the liver tissue, remember. Other fat means either it is converted into the non-essential amino acid, synthesis of the glycine from serine, and glutamine, asparagine are the form from the other molecule, okay, that is captured by the ammonia. Toxicity of ammonia. So one of the mechanism for toxicity of ammonia is affect the TCA cycle. TCA cycle affected, so ultimately decrease the ATP production, and that ultimately affect the cell. How it affect the TCA cycle? Increase the ammonia level. It ultimately bind with the alpha ketoglutarate and produce the glutamate. So ultimately alpha ketoglutarate, it is a consume, so decrease the availability for TCA cycle, because it is an intermediate of TCA cycle. So ultimately TCA cycle are the impaired and decrease the ATP products. Glutamate also, when ammonia level are increased, it is bind with the glutamate and produce the glutamine. So decrease the glutamate level also. So overall mechanism include the first, it affect the TCA cycle. It is the most common mechanism. Second, affect the potassium channel in the cell, neurons mainly, so affect the excitability. Third, by various mechanism it leads to the brain edema. Fourth, it affect the neurotransmitter metabolism. Most commonly NMDA receptor are affected. Fifth, various other amino acid metabolism are also affected. Sixth, alter the glutamate level, so it is required for the GABA production, so alter the GABA neurotransmitter level also. Glutamate itself, it is an excitatory neurotransmitter. GABA, it is an inhibitory neurotransmitter. So overall alter the level of the excitatory as well as inhibitory neurotransmitter and affect the various mechanism in the brain. So by that various mechanism, ammonia directly affect the neurons mainly. It increase the excitability as well as sometimes decrease the excitability of the neurons, and that ultimately leads to the unconsciously. So body is always try to maintain the low level of the free ammonia in the blood. So whenever it is a release from the any amino acid, it is immediately captured by the another molecule, mainly by the transamination mechanism. So free ammonia level should be kept less than 50 micromole per liter. If it is increased, then it ultimately affect the neurons and leads to the affect the consciousness, convulsion, coma, and various other signs and symptoms are the seen due to the higher ammonia level in the blood. Right? So that is a basic mechanism by which body, it is detoxify the ammonia and it convert the ammonia into the urea cycle. So whatever ammonia it is a release from amino acid metabolism, it is a enter into the urea cycle for the final detoxification. Thank you very much.